wasting my time.
So we're just gonna let it go away, and we're just gonna focus on 5, 6, and then the exam will be 5, 6, 7, 8. Yeah, that's enough. I don't like to add anything extra. Just a reminder, since we do have a quiz on Monday, which will take the first 20 minutes of class, we're gonna have homework due on Sunday. Remember, I use your homework as your study tool, right? So don't take the homework until after you've studied. You have until Sunday at midnight to submit it. Make sure that you submit it, but use it as a study tool. Once again, just like last time, there's no time limit. You can open it, you can go through and answer it, you can look at your notes, but it's better if you've already studied and you use this as a quiz for yourself, and then before you submit it, you double check to make sure you got everything right. Alright, so use that as your study tool. And then also the lab quiz, our typical lab quiz we have every weekend, it's going to be over labs 9 and 10. Alright, so we have 9 results on Monday and Tuesday, we have 10 results today and tomorrow, and so we'll go. And nothing on lab 12, which is what we're sitting on tomorrow for Thursday. Any questions? Yes.
Will the study guide still be posted?
I'll still post it. I'll probably get it up by Friday. Again, be careful with the study guide. Quizzes are more detailed and more in depth than the exam. So the study guide's a little overview, but I would study your chapters carefully, alright? If it's not on the study guide, doesn't mean it's not going to be on a quiz. You know, for the exams, I try to make them a little bit broader because it's a lot of information. But quizzes, it's only 2 chapters, I can't get specific ones. So I still will post it, but just use it with a grain of salt. Study guide is not meant for the quiz, alright? So just use it with a grain of salt. Anything else? Questions? Okay, so last time we met, we were talking about how we have physical requirements for bacteria and we have chemical requirements, right? What was one of the physical requirements?
Temperature?
Temperature, excellent. So we have to have good temperature. What was another physical requirement?
pH.
pH, excellent. And then what was the third one?
Osmotic pressure.
Osmotic pressure, great. We started talking about chemical requirements. So what are some of the chemical requirements? Carbons, yeah, making sure that they have an influx of constant vital molecules, food sources, energy, that they have trace elements, and then this, I believe, was our last slide, right, where we talked about cofactors. So this is our last slide. Okay, so other things that they're going to need. They're going to need organic compounds. Our carbon bases coming in all the time. The reason for that is we're going to need sugars coming in to make bases for our nucleotides. We already learned about this with which pathway? Which pathway makes DNA bases and RNA bases? The PPP, excellent, good job. So the PPP pathway that we covered in chapter 5 is what we're seeing here to make our nucleotides, alright? So we're going to make our purines and our pyrimidines based on breaking down glucoses and rearranging them, alright? So we're going to focus on that. Other things that can affect microbial growth is their oxygen requirement. So some organisms are like us, they have to have oxygen to survive, so we call those organisms obligate aerobes. So if we are talking about ourselves or bacteria that have to have oxygen, those are obligate aerobes. Now, a lot of our microbes, and probably the majority of them, are going to be considered to be facultative anaerobes. Now, we used that term facultative before when we talked about halophiles, right? Remember we talked about how organisms could turn on and off their salt pumps? They were facultative. The same is true with facultative anaerobes. They can live without oxygen, but they prefer it. And hopefully that makes sense why they would prefer it. If I go through aerobic respiration, so let's say oxygen is present, then from what we learned in chapter 5, how many ATP can I make through glycolysis, Krebs, and ETC for a prokaryote? Excellent. Oh, it's like music to my ears. Good. So I can make 38 ATP. If oxygen's removed and I'm stuck in fermentation, meaning I can't go on, how many ATP can I make? 2. Yeah, so if I don't have oxygen present, I can only make 2 ATP. Now for us, that's not enough. We die. That's why we can only last about 6 minutes without oxygen. Everything shuts down, we start getting brain damage, tissue damage, things are not good. For a single-celled bacterium, they can live on just 2 ATP for a while, okay, for quite a while. They don't love it, so if I give them oxygen, they're definitely switching back. handle a low reduced ATP load for a while. Now some microbes, they cannot handle oxygen at all, and we call those obligate anaerobes. So obligate anaerobes, oxygen is toxic to them, and we'll see why on a few slides. So with this idea of oxygen then, we can set up some new categories. So we have our temperature categories, right? Our mesophiles, thermophiles, all those guys. We can also divide bacteria into 5 categories based on their oxygen preferences or requirements. So on this side I have the organisms that oxygen is required for, obligate aerobes, and then this other group called the microaerophiles. These are ones that can handle and actually need oxygen, but they can only handle it in a smaller amount. So these would be organisms that need to also have like CO2 or nitrogen in their environment. If they get too much oxygen, it can actually be deadly for them. We'll talk about why. It has to do with their enzymes. Okay. Oxygen not required is for this side over here. These are the ones that can grow with or without, or that can grow without it. At the top, obligate anaerobes. So oxygen is toxic to them, it will kill them. Okay. Underneath that, in the blue box, facultative anaerobes. So again, these are the organisms that can go back and forth. Okay, they prefer it, but they can live without it. At the bottom are the aerotolerants. These are organisms that we think probably have been on the planet for a really long time. They evolved without oxygen being present. And then once oxygen showed up on our planet, they didn't care. So they just ignore it. So it doesn't hurt them, but it doesn't help them. They don't get any extra ATP if oxygen is present, but it is also not toxic to them, so they just ignore it. Okay, so the aerotolerants are just going to ignore it. And we're going to see why in just a second. It has to do with enzymes. So all of these organisms being put in these 5 groups, it's based on the enzymes they make. So at the top, we have oxygen. And oxygen, we're bringing in O2. O2 tends to get a little unstable with time. Oxygen, as we've talked about before, is an electron what? Electron high, yeah, he wants those electrons. So even when 2 oxygens are together, they still want to have more electrons. They're never happy, they always want more electrons. So what's going to happen with our oxygen is they can get reactive. And so what they start doing is these oxygens kind of start looking around. If they see cells or tissues nearby, they're like, you know what, I want that electron. They'll just go take it. They go grab it. And that causes damage to our tissues, to our cells. We don't want to have electrons going away. Luckily for us, we produce superoxide dismutase. So that's the enzyme that you can see right underneath the first damage there. Superoxide dismutase says, whoa, oxygen, slow your roll and calm down. We're going to give you a couple hydrogens, all right? So we give oxygen a couple hydrogens, and he goes from being reactive to making H2O2. What is H2O2?
Hydrogen peroxide.
Excellent, hydrogen peroxide. Is it good for us to have a lot of hydrogen peroxide? No, that can cause tissue damage too. So for us, we immediately have another enzyme called catalase that comes in and neutralizes the hydrogen peroxide to water and back to harmless oxygen. It reduces its state, it relaxes it. It's kind of like when somebody's hangry and you give them a snack. That's basically what we're doing. We're giving them a snack, we're calming them down, getting them to relax, and we have enzymes that are helping with this process the whole way. Now, some bacteria out there, they can also neutralize hydrogen peroxide, but they use a different type of enzyme. They use a peroxidase. We use a catalase, they use a peroxidase. It's fine, it just depends on which enzyme we produce. We only make catalase, some bacteria will only make catalase, other bacteria can make peroxidase. The end result is the same, I get oxygen in a harmless form. Okay, that's the end result. So, if I'm an anaerobe, the reason that I cannot tolerate oxygen is I don't make superoxide dismutase and I don't make catalase or peroxidase. So when oxygen becomes reactive, I have no defense. and oxygen starts ripping apart my cell walls, starts ripping apart my poor little like plasma membrane, everything gets ripped apart. So oxygen is toxic to me because I don't have these enzymes, okay? Now, for a lot of our organisms out there that are microaerophiles, so remember I said microaerophiles need oxygen, but not too much. So in that case, what they're doing is they're making superoxide dismutase. They get to hydrogen peroxide. They then have peroxidase that they're going to make, but they run into a little problem. They're slow making peroxidase. So these are going to be our microaerophiles. They can make a little of this enzyme, but not too much, which is why they need to not have too much oxygen present. So a lot of times we'll find these organisms growing deeper in the ground, where there's a little less oxygen in the soil. They're gonna, a lot of times, be growing in tissue areas that are a little bit more on towards the anaerobic side. They're still getting a little oxygen, but not as much. Okay, that's gonna be our microaerophiles. The way I like to picture microaerophiles, have y'all ever seen I Love Lucy? Or do you at least know who I Love Lucy is? Okay, she has a famous episode of I Love Lucy in the Chocolate Factory, where she's supposed to be boxing chocolates on a conveyor belt, and everything starts off okay, the chocolates are coming, but then about halfway through the conveyor belt speeds up, and she's like frantically like trying to put chocolates, some of you are nodding, yeah. That's kind of what's happening to our microaerophiles. I'm doing okay, and then too much oxygen starts coming in, and I have an I Love Lucy episode. I'm like, ah! And I start to freak out and die, okay? Because I have too much toxic oxygen near me. So whether or not an organism can live with or without oxygen, it's all to do with enzymes, all right? Now, another group that we talked about in there was our aerotolerant. The aerotolerants need oxygen, or they don't care? They don't care, yeah. So aerotolerants, remember, they're not hurt by oxygen, they don't need oxygen, so they're also producing these same enzymes. They're neutralizing it, but oxygen isn't their final electron acceptor, so they don't care. It doesn't impact them one way or another. Brandon, you had your hand up earlier.
Yeah, do we have to know all that?
I could ask you to explain to me why an organism has oxygen being toxic to them. So you could say something like along the lines of it would be enzyme based.
Okay.
We need enzymes to neutralize toxic forms.
Okay.
Okay, so usually I won't make you memorize the name of the enzymes, but I do want you to understand that it's multiple enzymes in the process to keep the oxygen from becoming toxic.
Okay.
Does that make sense?
Yeah, and then I had another question.
Go
You were
for it.
talking about like oxygen, like stealing electrons and stuff. I've heard the term in like nutrition, like videos and whatnot about like free radicals and like oxidation and stuff. Is that what that is?
That's exactly what that is.
Okay.
So there's certain drinks or foods out there that are antioxidants, right? And those are super healthy. What those are doing is they're neutralizing those radicals. They're neutralizing that superoxide.
Okay.
They're calming them down. Okay, so that's what those healthier foods are doing. They're preventing damage.
And is there things that like you can eat that are like more inflammatory or like more that cause more free radicals?
A lot of times it's genetics, how much of an enzyme we're making. So that's why some people are more prone to issues than others. Sometimes it's diet. If you're eating a well-rounded diet, you tend to have enough enzyme present and enough hydrogens present to neutralize the oxygen. But if you're eating a poor diet, maybe the dietary issues aren't meeting what you need.
Oh, okay.
So it can cause inflammation, things like that. Also immune systems. Some people have immune systems that target themselves. So we'll talk about that later in the next chapter.
Okay.
14. Okay, so is this good? We just understand it's all enzyme based. So this is a way for us to tell oxygen preferences in different bacteria. And this is a special type of broth. We'll set this up in lab soon. That's called FTM, or fluid thioglycolate media. Ooh, that's a mouthful. FTM media. FTM media is a broth that is a liquid. It looks like a normal broth, but the key to FTM is it has a little agar in it. Just a little, not enough to solidify it, but enough to make an oxygen gradient. So if section B. I see that at the top, I have zero growth. No growth at all. So what does that tell me about the organism? Does he like oxygen? No, he's hiding from it. He's hunkering down here. So that tells me that organisms are obligate anaerobe, not able to handle any oxygen. The next guy over, so in C, I have a facultative anaerobe, and here's how I can tell. I have a lot more growth where oxygen is present. It's a little thicker, it's almost like a mushroom, but as I move lower, I'm still growing. I prefer oxygen. I grow better where there's oxygen, but I can grow without it. So that's the sign of the facultative. Next one over is a microaerophile. I want some oxygen, but if I go too low, I die off. I can't live without it, but I can't live with too much. And then at the end would be us, the obligate aerobe, where we're all like in a swimming pool gasping at the surface. All right, and we don't want to get pushed under that pool because we're going to drown and die. So that's what's happening with our obligate aerobes right here. Okay, so if I have a bacterium and I'm like, gosh, I wonder what this is. Is it an aerobe? Is it an anaerobe? What is it? I can inoculate it into FTM, and based on its growth pattern, I can tell what its oxygen requirements or preferences are. It's pretty cool. It's pretty cool way to do that. And we are going to have a lab where we're going to do this. So, some microorganisms, we mentioned this last time, this is a throwback, use gaseous nitrogen, so N2, directly from the atmosphere. And so they're able to pull that in and convert it to a usable form for plants. Was that called chemosynthesis, photosynthesis, nitrogen fixation, or nitrogen synthesis? C, yeah, nitrogen fixation. So they're fixing nitrogen that is unusable for most organisms, and they're fixing it into a usable form. Okay, so they're going to convert it. This is the one I told you about where the plant roots will send out sugar to try to trap the bacteria, and then they grow together. This organism is killed by atmospheric oxygen. I heard it. C, obligate anaerobe. Good job. How would I determine whether a microbe is a strict anaerobe? What could I possibly use in my laboratory? My FTM. Yeah, I said it, my FTM broth. And based on where it grows, I'd be able to tell whether it was an obligate aerobe, obligate anaerobe, microaerophile, any of those. So FTM's always the way to go. And again, that stands for fluid iobiovolate medium. If I had that on a test, I would say FTM, by the way, because that's molecule. Okay, are we good to go to the next slide? Okay, all right, so let's start talking a little bit about culture media. This is kind of nice because this, again, kind of like what we had in chapter 3, it starts to go hand in hand with some of our lab stuff we've been covering. So, as we mentioned in lab, culture media is basically where I'm giving nutrients to bacteria to be able to grow them. So I'm giving them things like biomolecules that they need to survive, and that allows them to grow and thrive on the media. Now, to use my culture media, it has to be sterile. So we have to make sure that there's nothing on there before we start. Right? We don't want to have any colonies already growing there. And then when we get our sterile media, we're going to add bacteria to the media, and when we add bacteria, we refer to that bacteria as our inoculum. We're inoculating the plate, that's the term we use, and we're putting bacteria in the plate. We're going to let the organisms grow, and when they grow, we refer to that as a culturing. So one of the jokes in microbiology is microbiologists have a lot of culture, all right, because of the fact that we're always culturing, right? We also talk about how we do a lot of scripting. That's probably not as friendly. Yeah, because we do a lot of script plates, right? So they have t-shirts for microbiologists that have all these nerdy terms on them, and we all love them, and we all get them, because that's how we are. Okay, so we also, in our solid media, use agar, and we talked about agar in lab as well. So this is kind of review. It's a polysaccharide, so that means it's what? A carb, a protein? Carbohydrate, good. It's used as a solidifying agent, and the reason we use it is, as far as we know, nobody can eat it. And I always put a little caveat to the side, because I swear, every biology principle out there has one exception. We have not found that yet. That doesn't mean it doesn't exist. There's probably some bacteria out there making race, but we haven't found it yet. This is way better than gelatin peptone, where everybody can eat gelatin, or most everybody. So agar liquefies easily, why we really prefer to use agar now as the farm agent. Before agar, as I mentioned, we had gelatin. Gelatin would break the hearts of Robert Koch and early microbiologists because it would start nice and solid, but if I had an organism that produced gelatinase, it'd come in the next day and see a swimming pool. So that was just kind of disheartening because they were trying to see individual colonies, and instead they had these melted petals. And that was because the organism could eat the gelatin. So finding agar was a huge, huge plus. We wouldn't be where we are today without it. Now, as far as culture media goes, we have a couple of different types of our sleeves that we can use. The first way that we separate culture media is into two distinct groups. The first group is chemically defined media. This is where we know every single ingredient in the media. And you can find, like, if you get culture media is sold like instant cake mix. You get it in a box, it's powder. You add your powder, you add your water, and you're done. That's actually how we make our petri dishes and our broths. It's like all done behind the scenes, but it's like cake mix batter. So in this case, your cake mix batter, every single ingredient is known in it, okay? Now, chemically defined media is expensive. It's a lot of money to know every ingredient in there, and it's very precise. Usually, we don't need to be that precise. So what we tend to use more than chemically defined is a group of media called complex, which is this guy. Complex media means I have some carbohydrates and some lipids. I don't know the exact amount, I just know that there's some in there. So for example, it's kind of like when we go shopping at the store. We buy some maybe ground beef, and we know that it's, maybe it says on there it's 85% lean. Okay, so we know roughly how much fat's in there. But do we know how many carbs are in there? We don't know what that animal ate. We don't really know anything about it. It's just kind of a rough guess, right? That's what complex media is. It has all the stuff you need to live, but we don't know the exact percentages. With chemically defined, we know the percentages. So the real differences and the real reasons we use these two is this is always our first choice because it's cheap. This choice is only if we want to reproduce results without any variation, okay? In other words, we're holding everything constant in the plate, and the only variable is the bacteria itself, okay? That's the only variable. So when that becomes important is going to be when we're doing something like antibiotic testing. If I'm testing antibiotics, I don't want to have a plate that maybe because it had more lipid than this other plate is going to have a different growth pattern. So I'm going to have to know every single thing in it. That way my only variable is maybe the antibiotic itself for the bacteria. So here's an example of chemically defined. Every single ingredient in here is known. We know the concentrations. We could calculate how many moles of hydrogen are here, how many moles of oxygen are here. We know everything. There are no variations. Complex media, again, is going to have extracts, and I'll show you a picture of that. So, chemically defined, we know everything is important with things like this. This is antibiotic testing. So the way this works is each of these discs have been soaked in a different antibiotic. And what we did is we set up a plate with a whole bunch of bacteria, one bacteria grouped all over the surface of the plate, and we come in the next day and we look to see which antibiotic is killing it. So the way we determine that is we measure what we call zone sizes. So if I'm a patient, do I want antibiotic C? No. Antibiotic C is not trustworthy, right? So that makes us feel a little nervous when we see something like that because that bacteria is completely resistant, right? This is kind of like Staphylococcus aureus. What was the very first antibiotic ever discovered?
Penicillin.
Penicillin, excellent. We unfortunately used penicillin so much, staph became resistant. If you put, and we actually did this when I was a student in Cal Poly, we ended up getting a collection of bacteria from the 60s. It was pretty cool. It was from a researcher who retired, and he sent us some of his freeze-dried organisms. And we grew these organisms, and when we put that staph onto a plate with penicillin, it had a zone like this. That's from the 60s. And then we took a sample of staph from the back of your tank get you that way. I mean, that's the microbe philosophy. So again, this media is really, really expensive, but I have to know that there's no variations because I need to make sure the only variation is the antibiotic. Does that make sense? Why chemically defined as used? Now, extracts or composition of complex media, it looks like this. It has some beef extract. Well, that means that I have some beef in there. I don't know. Maybe it was a really chubby cow. Maybe there's more lipids. Maybe it was a really skinny cow. Maybe there's less lipids. I don't know. It doesn't matter. As long as there's a few lipids, my bacteria are happy. I'm not going to see enough variation to make a difference. Does that make sense? So from plate to plate, I might have 20 grams of lipids here. I might have 18 grams of lipids in this Petri dish and 19 in this one. Does that matter? No. The bacteria don't care. They're going to grow the same. It only matters if I need to make sure everything's perfect, like antibiotic testing. Make sense? So it can be a little bit cheaper to make this kind of media. Okay, so complex media is great because it's cheap, it's fairly easy to make, I can grow a wide variety of organisms, and I can adjust it and put different ingredients in it to make it special. That's what we've done with things like mannitol salt agar. Mannitol salt agar is complex media, and then we added reagents to make it selective and differential. So it's like taking that cake mix, and now I can add different colored food colorings and different things to make it special. Okay, that's all I'm doing with the media. Now, another thing that we can refer to with our media is that we can say that some media that we purchase has been reduced. So reduced media is basically going to be media that has been prepared in a room where there wasn't oxygen. So it's either been maybe put into a heated room where the oxygen was pulled out, or some sort of a vacuum where there's not as much oxygen trapped in it. And the beauty of this is it's trying to make sure that the media is as anaerobic as possible. So for example, FTM, when we purchase it, it's been reduced. So what that means is when they make the FTM, they boil it in the tubes, and then as soon as it starts to cool a little, they tight cap. And that prevents the oxygen from coming in. So when we boil the oxygen left, we tight cap it, and then it's sent tight capped. And so the only time oxygen enters is when we open it later to put bacteria in it. That keeps the bottom part completely anaerobic. Alright, so these are special media that are used just for our anaerobes. Now, let's say I want to grow anaerobic bacteria, but I don't want to use a broth. I want to be able to spread them out and see the colonies. So if I want to grow anaerobic bacteria on a plate, I can do so through a couple of different options. We're going to do this. We're going to do a lab where we grow anaerobic bacteria in FTM. We're also going to grow some anaerobic bacteria on a plate. And what we're going to do is we're going to set it up, exposed to oxygen, and then we're going to put our plates inside of this special jar, and we're going to add a packet in there that's going to pull and trap the oxygen out. So when that happens, we're creating basically a little mini anaerobe chamber, a little chamber that has no oxygen in it. So my anaerobes will be able to grow in there. Now you might ask, well, wait a minute, what about when we're first setting them up? Well, unfortunately, when we're first setting them up, they're dying. They're exposed to oxygen, they're not happy. But luckily, most obligate anaerobes are going to be able to make endoscopes. Because they understand their life is hard. They're going to get exposed to oxygen. So they have endospore capabilities to be able to survive until it's anaerobic again. Alright, so they're able to survive while we not really. If we want to get really fancy, we can use anaerobe chambers. So I have a different picture. You guys just have an empty chamber. I have a picture of somebody in one. So an anaerobe chamber is where you have a box that's sealed. You have a pass window. And the way this works is this window gets flooded with CO2 or nitrogen or some sort of gas that's not oxygen. And this is where you put all your samples and everything is in this passport. And then what you're going to do is you're going to put your hands in these gloves, and after you've flushed all the oxygen out, you open this little port valve and you pour your products and everything inside. Then you quickly close it, and then the bacteria and everything you're setting up are exposed to completely anaerobic environments. So it never gets exposed to oxygen. So my bacteria, everything that gets passed through that little window in a FTM broth, they never get exposed to oxygen. This is used in like places that are doing research on anaerobes. You might see them in a couple hospitals. It's kind of on the rarer side because, as you can imagine, it's expensive. And you have to have gas tanks nearby, which could be knocked over, and it could be a safety concern, right? Because anytime I have a big pressurized gas tank, that can be a concern. Capnophiles. Capnophiles are a little odd group. We haven't mentioned these guys before. Capnophiles are going to be organisms that really like a lot of CO2. Okay, so they can grow with oxygen. That's fine, they'll go kind of slow. Their preference is CO2. So if I had a capnophile in my body, where do you think I would find it?
Lungs or circulatory system?
In the lungs, yeah, and in the circulatory system where I have a lot of CO2 coming through. So tuberculosis is considered to be a capnophile. It can live in oxygen, that's fine, but it's way happier if it has more CO2. Okay, so if I have a CO2 preference, if it helps you grow faster, then that makes me a capnophile. So that would be, the vectors I see these would be like in the lungs primarily, because they have a lot of CO2 coming through. Okay, selective media. So we talked about this in lab 2. Selective media is going to be media that is going to kill all organisms that we don't want to grow. That's the big thing about selective media is I kill everybody off. I am then going to encourage the growth of the organisms I do want. Okay? And we've already used some selective media. We've used MSA, which is mannitol salt agar, which is the one you see here. We've used McConkey agar. Sabdex is a special agar for yeast. And remember I told you yeast can handle low pHs. So Sabdex tends to be a more acidic agar. So the bacteria die because they can't handle the pH. So selective media means I'm selecting the organisms. Differential media. So differential media means that I am seeing a difference between the organisms that are there. Now, the thing about differential media is so far in our lab, when we've seen differential media, it's been hand in hand with selective. They don't always have to go together. So for example, right here is a blood agar plate. We talked about how we get blood agar from sheep, right? We also can get them from horses, from cows, from all different animals. Usually 9 times out of 10 we use sheep's blood. With this sheep's blood then, in this plate, when we set up different bacteria, they're going to show a different growth pattern depending on the enzymes they make. Now this media is unusual because it is not selective. It'll grow everybody, but they will look different based on their enzymes. Okay, so let's look at this. First of all, there's alpha hemolysis. The way I like to think about blood plates is we're looking to see if the organism can eat red blood cells. Okay, would we be worried if an organism can eat red blood cells? Yeah, that's a sign of a pretty big pathogen. Okay, so when you picture your red blood cells, I want you to think of a hard-boiled egg. Okay, and I'm going to explain why. First off is alpha hemolysis. This is where we have pathogens that basically are taking a hard-boiled egg, we're pretending that's our red blood cell, and they're cracking the shell. Okay, they're damaging the red blood cell, but they're not able to completely break it. They're not taking the shell off and eating the egg. Okay, so they're cracking it, they've dropped the hard-boiled egg, the shell has been broken, they're damaging the red blood cells, but they're not fully breaking them apart and destroying them. So that's what we see here with alpha hemolysis. The blood looks a little greenish tint. That means the red blood cells have damage. They've been hurt, but they have not been destroyed. Okay, so they're damaged, they're hurting, but I haven't ripped them apart. This is beta hemolysis. Beta hemolysis is a whole different game. Beta hemolysis is where I'm breaking the shell in the hard-boiled egg and then I'm eating it. And I'm eating the shells too. I'm gonna eat everything, all right? So beta hemolysis, and we'll do a lab looking at these microbes too. If I take this blood plate and hold it up, normally blood is opaque, I can't see through it. Where I have a beta microbe, it looks like a window. I can like hold it up and see right through it like glass. Now, there's an organism that is commonly known for beta hemolysis. You guys might have heard of them, strep throat. Streptococcus pyogenes. Anybody in here ever had strep throat? See some nods, yeah. If you've had strep throat, you know the pain of this, right? It hurts to swallow, it hurts to talk, it hurts to drink, it hurts to eat. And the reason for that is because they are destroying, they're clearing any blood cells and tissue that they can get in contact with. These are also, same streptococcus are also known to cause toxic shock. They're also known to cause heart damage if they're not treated. So there's a lot of diseases with that streptococcus strain because it is so aggressive and so rough on our tissues and our blood cells. The last little group down here is the gamma group, and they are completely harmless. They're just looking at the hard-boiled eggs but not touching them, all right? So they're not cracking them, they're not hurting them, they're doing nothing. So these down here, they tend to be pathogens that are not as damaging, if they're pathogens at all right? So they tend to be just completely not damaging. So that's a great example of a differential plate that is not selective. Okay, anything will grow and blood. Okay, the last one we'll talk about is the enrichment media. And enrichment media is where we are encouraging the growth of a desired organism, but the key difference is we're not killing anybody. Okay, so I mentioned in lab, this is where it's like your favorite foods, your favorite drinks being given to you to encourage you to grow. But I don't have anything toxic in there to kill any of your neighbors. Okay, so these a lot of times are used when we suspect we have a pathogen, let's say Salmonella or Shigella, but we're not 100% sure. So we're going to give food that Salmonella and Shigella love, but I'm not going to kill anybody else just in case I'm wrong. So maybe it's 0157. Maybe it's a different Proteus strain. I don't know. So I don't want to kill anybody in case I'm wrong. I think it's this, but we'll see. So this is also used in environmental media. Let's say I have an organism that can break down plastics. So I'll grow it in broth that has plastic components, and then I'll see if it can eat them. But who knows? Maybe instead of plastics, I'll see somebody breaking something else down. So I don't want to kill them. I just want to see what else is there. Okay. So the big difference between selective and enrichment is that I'm just encouraging growth with my enrichment, not killing. Selective, I'm killing. I don't want anything out of the background. Now, media is great. We all love media. The problem is there are some microbes out there that are truly the princesses of the microbe world, and they will not grow on fake media. These pathogens want a living host, all right? So they are not going to grow in anything else. One of the big ones, Mycobacterium leprae. What do you think that caused? Leprosy, yeah. So Mycobacterium leprae, he's been renamed. He's now Mycobacterium pneumoniae, I believe. That's because the stigma associated with leprosy, so they're kind of changing his name. But we're still going to learn him this way because it's easier for us to remember. Mycobacterium leprae is an organism that we can't grow very easily. We have to have living organisms. So it turns out that we grow this organism in the heel pads of armadillos. How they figure this out, I will never know. I just picture them bringing in a list of animals that start with A. And they're like, okay, let's try the antelope. Nope, it didn't grow in the antelope. Let's try the oddball. No, it didn't grow in the oddball. Bring in the armadillo. Oh, it grew. Okay, we can stop. Thank gosh they got through the A's, right? Thank heavens it wasn't like a zebra. Because think of all the animals they would have lost along the way, right? So in the heel pads, the little paws of armadillos, they'll inoculate them with Mycobacterium leprae and watch the bacterium grow. And then from there, they can take samples and they can do what they need to do. But if they try to use a dog or a cat, it doesn't work. Different biochemistry. Who knew that we were so close to armadillos, right? That makes me kind of question my life, but I'm not going to think about too much because I just want to move on. Okay, Treponema pallidum. We've heard of him before. What is it called? Treponema pallidum?
Syphilis?
Syphilis. Good, yeah, syphilis. Good job. Treponema pallidum. If I try to grow syphilis outside of the body, it is a no-go. It is not going to grow on agar. How dare I give it agar? Oh, what am I, common bacteria? I don't think so. Treponema pallidum is grown specifically in male bunny rabbits in the testes. And I know, we feel sorry for the bunny rabbits, we thank them for their contribution. But yeah, they are grown in rabbit males in the testy tissue because they won't grow anywhere else. They tried the armadillos, it didn't work, so they had to bring in the rabbits. So just kind of some weird things. There's a few other microbes that are out there that have to be grown in animals. We try to not do too much animal testing, but unfortunately sometimes we have to because there's no other way to grow the bacteria. Okay, a checkpoint. I'm going to cultivate an anaerobic bacteria. I can use a special media. What would my media be that I'd want to use? Reducing, good. So remember that means that I have removed the oxygen from that media as much as possible. Okay, the next one. The culture medium that is designed to increase numbers of desired microbes to detectable levels. Now no, I'm not saying killing. What would that one be? Enrichment, C, good. Now if I said the same thing and said, and I'm killing the background microbes, that would be which one? Selective, good. So I want to make sure that we understand that difference. Selective means I'm killing all the background. Okay, so what about our microbes that we grow? Well, as we learned in our lab books, I have it in the very front of your book on page 5, we have a list of the microbes that we use in our labs. And I have them separated into BSL1 and BSL2. And I told you we would talk about what a BSL was in lecture one day. Well, that day has arrived. So a BSL is a biosafety level. Every microbe and virus out there is evaluated to see how hazardous they are, how likely they are to make us sick, and how easily they can be passed from person to person or through the air. And so every organism that we work with is given a BSL level, biosafety level. BSL1s are the lowest risk. No special requirements. These are the things you're going to run into at the grocery store on your shopping cart, at the door handle getting in and out of your door. These are microbes that are just background. So these would include things like E. coli, Staphylococcus epidermidis, Bacillus megaterium. That's another one that's found in soils. We've been working with Bacillus subtilis. It's very similar to megaterium. Totally helpful organisms, a lot of times not harmful at all. They're not easily spread. Now that sounds weird because you're like, well, if I touch the door handle, I'm picking up the microbes. What we mean by that is, let's say I get a rare infection with E. coli. Maybe I have a UTI. Can I spread my UTI? No, I really can't, right? It's hard to do that. So if I do get an infection with it, I'm not going to be able to easily spread it. Okay, let's go to the next one. BSL2s. BSL2s are where we're stepping up a little. We want to have some precautions for certain organisms. So in our lab, we wear lab coats. We wear gloves if we have open wounds, but we tend to wear gloves anyway. For certain organisms, if we're vortexing, which means shaking, we'll wear eye protection. We don't do that. We never really are shaking at a high speed, so we don't have to worry about that. If I do get sick from these microbes, my disease is going to be super mild, okay? And it's going to be easily treated. Now, you might be surprised to see the list of organisms that are considered to be BSL2s. Are y'all surprised by that list? I know, like we kind of look at that like, so I said that their diseases could be mild, but another key thing is they're not easily acquired, not easily spread. So let's look at this guy. Let's start with MRSA. So MRSA is methicillin-resistant Staph aureus. Well, it turns out Staph aureus infections, you don't just pick them up just by touching them. I have to have an open wound, or I have to have some way for the bacteria to really get inside. Because what are my hands normally covered in? Staph epi. And if Staph aureus tries to come in, Staph epi's like, whoa, buddy, this is my body. You get out of here. And it'll compete with its own cousin. All right, so Staph epis usually keep the Staph aureus away. However, if I get a cut or a wound, that Staph aureus can get inside. Okay? Methicillin-resistant staph, all it is it's a typical Staph aureus with an enzyme that neutralizes some antibiotics. Don't worry, we can treat it with other antibiotics, so it's still treatable. If you've ever seen MRSA, if you catch it early, it's a quick treatment. It basically looks like a little spider bite. It's a spider bite that doesn't go away, and it gets very tender, it gets very sore. Your doctor will go in, they'll pop the spider bite, remove the pus, pack it with some antibiotic, and send you home. It's a real quick treatment, and you're done. Okay, next, Lyme disease. Ooh, we hear about Lyme disease a lot, right? It turns out most people who get Lyme disease don't even know they have it. What does Lyme disease come from?
Ticks.
Ticks, yeah, it's carried by ticks. A lot of people who get Lyme disease have no signs, no symptoms, and no further complications. It seems that unfortunately there's a small portion of the population who their genetics do not work well with Lyme disease. So when they get Lyme disease, they start to get inflammation responses. And it's actually our immune system response that starts to lead to damage. We start to have memory loss, we start to have joints hurting, tissues aching, because our immune system's kind of overreacting. And so Lyme disease on its own, it's actually pretty mild in about 90% of cases, which is really shocking. Influenza. Influenza, I don't know about you guys, when I get influenza, I feel like crap. I get really sick, right? But how long are you sick for? A few days. Yeah, a few days. You need to get over it. And then also, influenza is something that tends to be fairly difficult to contract. As weird as that sounds, I have to have a pretty high viral load. So I have to be near somebody when they're early in the flu and they're coughing or sneezing and they're having a lot of virus release. Because if I get somebody on the tail end, their virus load is too low, I'm not getting hit with enough viruses to actually get sick. The last one, HIV. Another one, it's like, wait, because what does HIV cause? It can cause AIDS, yeah. But it turns out HIV is actually kind of tricky to acquire. Like you can work with HIV on a lab, the only risk that you can acquire with HIV is if you stick yourself with an infected needle. That's really how most healthcare professionals get exposed, is through an needle stick. But I could work on it on a lab all day, and it's not going to send the jump on me. All right, so kind of weird, I know, but it's because of the way that it interacts with our bodies. The next biosafety level, now we're up in the ante. Biosafety level 3s. These are where we no longer want to work on our benches, like what we usually do. Instead, we want to be working under a hood. We call these a biosafety cabinet. Now the way hoods work, and we have a couple, we have one in the corner of A105. You turn the hood on, you have your bacteria in there, and when you're working, it's blowing air away from you. And the reason that's important is these are bacteria that are small and light, and they can go into the air. And so if I'm working on a bench like this, I could breathe them in, and then they can make me sick. All right, so working in a hood keeps me from breathing them in, all right, keeps me safe. So BSL-3s, we're going to use exhausted air. It doesn't recirculate, meaning it doesn't blow back at you. It goes through a filter that's going to remove the microbes. You're going to want to wear some scrub suits or some coveralls, something a little bit more than just a lab coat, like what we wear. And it again requires a BSC, that's a biosafety cabinet. Biosafety cabinet, that's just the hood that you're working in. Now, the diseases here are a lot more serious. They can cause more damage, more discomfort, but luckily there are vaccinations available and treatment available for all these. So, for example, tuberculosis. You can get exposed to TB working in a lab. It's easy to breathe it in. It has happened many times. Did I tell you guys my story of when I was training in public health for TB? One of the guys that was training me, I love this guy. He was like 62, 63, old curmudgeon guy. And I just loved him because he knew everything. If you can find an old microbiologist, you're going to learn the best. We're going to teach you the old stuff. He would look around when he was getting ready to set his TVs up. He'd have the hood on. He'd look around and he'd light a cigarette. That was his smoke break. He'd be smoking while he was setting up TVs because the air would pull the cigarette smoke away and our manager wouldn't know. He never converted, never became positive. He did this for over 25 years. He'd have a cigarette hanging out of his lip while he's setting stuff up, and then he'd like blow it into the safety cabinet, crack it up, and never got it. I mean, his technique, it worked. So it's pretty cool. But if he hadn't been careful, he could have contracted the illness. And do we have a treatment for it? Yes, but how many months does it take? 6 months, so it's a long treatment. Bacillus anthracis, again, anthrax, easily aerosolized, I can breathe it in. If I have any wounds on my hands, I can pick it up, it's nasty. I can treat it, it's just gonna take a while. West Nile virus. West Nile virus is one that tends to be more reactive with people, so we want to be a little cautious with that, it can make us pretty sick. The reason we really worry about West Nile is it can have high fevers, which can cause brain damage. That's really why we worry about it. And Salmonella typhi. If you've ever had food poisoning by Salmonella typhi, you'll know because you're going to have about 5 days of misery. You're going to be super dehydrated. You're going to feel like crap. Your stomach's going to hurt all the time. You might even have bloody diarrhea. It is not a fun illness to get. You do not. Okay, biosafety level 4. This is where the crazy rock star microbiologists work. When we see these guys at conferences, we're like, we're not worthy to talk to them because these guys are crazy. They work with things that will kill you. They work with Ebola. They work with Marburg virus. They work with Lassa fever. These are all what we call hemorrhagic viruses. What does hemorrhagic mean?
Blood loss.
Blood loss, yeah. These actually will liquefy your tissues in your organs from the inside out. One of the signs of Ebola is you start having bloody tears, blood coming out of your ears, blood coming out of your mouth. Any orifice, you're going to have blood coming out. Not fun. You are being liquefied inside. It is easily transmitted. If somebody has Ebola and they're sweating and I touch them, I can pick it up. It is that easy. It is a nasty illness to have. Same with Marburg, same with Lassa. Now, the way these work then is we're not going to have you working in just a safety cabinet. We're going to have you working in a separate room. Your room is going to be key coded and there's going to be a lot of protection with that room. A normal microbiologist can't go in there. You have to have a special clearance. Kind of like on the base. You have to have clearance to get to that room. Once you get into that room, it is negative pressure. Negative pressure means that there's basically a vacuum in that room. If a door is left open, which it wouldn't be, but if it is, it's going to pull dirty air in rather than risk having the air go out. Okay, so that's pulling air into the room. The organisms that we work with class 4s are not treatable. So if you get it, good luck. If you survive, that's good genetics and you're okay. If you don't survive, that's on you. That's just what happens. There's no vaccinations. There's nothing I can give you to help prevent that. We're just going to have to do a wait and see approach. So the way this works, you put on a big bunny suit before you go into the room. You'll be inspected for any tears or any issues with the suit. You go in, you do your work. They pump oxygen into your suit. That's how you leave. You put a little astronaut. When you're done at the end of the day, you leave. You walk through bleach solution. You take your suit off. You get sprayed down by bleach. You then take your suit off. If any tears are found, you're going into a 2-week quarantine. You're not leaving the facility. You'll probably get to call your family the next day, but you're going to get isolated immediately. Yeah.
Is it true that many viruses are dealt with in space?
A lot of viruses are dealt with from here. Are dealt with in BSL-4.
But are any dealt in space?
Are there any viruses in space? That's a good question. We don't know. And I'll be honest with you on that one. The reason we don't know, and I can't say for sure, is there was this idea that viruses came to the planet on meteorites. But the heat with re-entry, we don't think a virus would really be able to survive that. We're not sure. Now, can I take a virus with me out into space and work with it on the space shuttle? I don't really want to, because even though I'm in an anaerobic area, so to speak, what's going to happen if a vial breaks? There's like a fluid floating around, and hopefully it doesn't bump into anybody. Now, let's say it bumps into them. Oh, sorry, now you got Marburg. Yeah, so I don't want to do that. Oh, sorry, now you got Ebola, now it's going to get messy. Now there's going to be drops of blood everywhere. So it's something I've seen in science fiction novels, but it's not something we do. But we do bacterial studies. We will take up BSL 1s and 2s, and I'm sure they've taken a BSL 3 even though they deny it. And they'll see how it reacts on the petri dish. They'll monitor the growth, they'll check it out and see what it does. They'll see if it mutates, they'll bring it back and regrow it to see if it has any genetic changes. So they will do that. But nothing is pathogenic as this. There's a big debate about COVID. There's a lot of information that's come out that makes people suspect that COVID was accidentally released from class 4, from BSL-4. And the reason for that is COVID was being studied, and it's a natural disease that occurs with bats. Okay, so it's a coronavirus that's found in bats. Now, where COVID was first released in China, there's a class 4, a BSL-4, right in that same neighborhood. And so the question that a lot of scientists have asked is it possible that something happened, a tear in a suit or something that wasn't caught, and then COVID was transferred to that person and then they accidentally not without any knowledge, but took it out into the environment. So that's something that's been proposed by a lot of healthcare workers. It is something that is not popular because it feels like it's pointing fingers, so people get nervous, but there's a lot of data that seems like that's a possibility, right? So biosafety level 4 is very dangerous.
I think I heard that the lab was called the Wuhan coronavirus lab or something.
It was. They were studying, it's very odd. They were studying coronaviruses, they were manipulating the genes to make them more mutacious and more deadly, but they didn't do this.
Yeah.
It's kind of like,
Coincidental.
just randomly happens. It gets a little suspicious. It's a little, my daughter would say it's a little sus. So, it's something that, again, when we kind of realized what was happening, they tried to send somebody to the lab and they were denied access. They were finally allowed in the lab almost a year later, and the lab had been shut down and that area was bleached. So there was like, and all the records were lost. So it's a little thing, little sus. So we're lucky though, because COVID, if it did start as a BSL-4, through its mutation, it has definitely dropped. So COVID comes in closer to a BSL-3 and sometimes even a BSL-2, depending on what mutation it is. So it's passed through aerosols easily, but it's not quite as deadly as we know on these other ones. So have you worked with four? Have I worked with BSL-4? No, I don't do that. I went to BSL-3s, and that was it. I was like, yeah, if you wanted to go to BSL-4, it's a whole different ballgame, and I like to live. I don't want that kind of stress. It just scares me. Plus, I don't know if y'all know, I'm klutzy. I trip over biohazard cans. Can you imagine me at a BSL-4? I'd be like, oh, scan myself, like, oh crap, I gotta go into quarantine again. I'd be in quarantine every 2 weeks. It'd be like, there's Hadley sitting in her little lone cell waiting to see if she'll die. That would be me. Okay, so here's a picture of a BSL-4 lab. So as you can see, this is serious stuff. I mean, these scientists, they are completely gowned up. They have these little shields over their heads. These hoses coming in are pumping in oxygen to them so that they can breathe. They're not breathing anything, and they're in the atmosphere outside, and they're even still working in safety cabinets. So I mean, this is like very big deal. And to get into this lab, you would have to have a key code clearance, and there'd be multiple rooms that you would go through to even get here. So yeah, I did not want to go in BSL course. Yeah, those guys, there would be adrenaline junkies. They get excited about things that kill you. So here is just a quick breakdown then. BSL 1, 2, 3, and 4. Just kind of going through a reminder of some of the diseases that are out there that are in each field, and the different steps that you can do to minimize risk. Okay, so really, if you think about BSL 1 and 2, think of the lab, think of what we're doing there. BSL 3 is a step up, you're on a hood, and you need to have UV vaccinations to work with some of the organisms. But if you get it, you can still treat it. BSL 4, no vaccinations, no treatments. You're working in a special lab, you have oxygen being pumped in, that's the one where if things go wrong, you're in trouble. Okay, let's talk about obtaining pure cultures of our bacteria. So when we're growing our bacteria, as we mentioned before, our goal is always to make sure that our bacteria are isolated and that we know what microbes we're growing. So the way that we're going to do this is by using pure culture techniques. This is where we only have one species or one strain of bacteria present. When we put bacteria onto a petri dish, we're looking for CFUs. CFU stands for colony forming unit. We want to see a colony, which is simply a single cell that has started to replicate and produce its own little population. Okay, so that little colony is supposed to be cells that are very similar to one another. Maybe I have a couple mutants in there, but for the most part, they're homogeneous, right? The way I get my nice isolated cultures is to do the straight plate method. We did this, right? With Serratia E. coli, just like what we see here. The Serratia produced those red colonies. E. coli was that more yellow or white. And here we have on the street plate the isolation, so we can find those colonies that are safe by themselves, take a sample on a lid, and then do additional testing of those. All right, can you think of a reason when we grow our colonies on a petri dish that the neighbor nearby is gonna be like, whoa, buddy, stay in your lane. You can't come over into my side, all right? So having a limited amount of resources, I can only put so much food and nutrients in a petri dish or in a tube, and then lots of competition. We're all fighting for those nutrients. Kind of like the housing market today. Lots of competition and it's a little frustrating. Same is true. Could a pure culture of bacteria be obtained by a street plane method? If I was only looking for one organism, and that suspension had brilliance. So in other words, I'm looking for a needle in a haystack. Can I do that with a street plate? I actually can. If I use a street plate, and if I can separate them out enough, I can find my needle in my haystack. That's what microbiologists do when we're doing samples from patients. We have selective media that can help, we have differential media that can help, but we're going to be trying to isolate and separate those colonies out, and we're going to be picking out the pathogens, those needles in the haystack, with all of the background. And that's what you have to study microbiology for years, is learning what they look like, the characteristics and microbiology. Okay, let's talk about preserving bacterial cultures. Let's say that I have some bacteria that I really like, and I want to save it. Remember I told you about the 1960s Staph aureus. So that was a culture that had been preserved, which was really cool because we got to play with it with penicillin. The same is true if I have maybe a hospital that has an unusual patient sample. They'll save it. They'll send it to the CDC, and the CDC, what's the CDC stand for? The Center for Disease Control. They'll do analysis and try to figure out if this is a new strain or if this is a mutation. So preserving cultures is helpful. We can preserve bacteria a couple of different ways. One of them is deep freezing. So this is where we put bacteria in a special broth that is going to preserve them at really cold temperatures. This is also what we do with special tissues. This is also how we do IVF. It's where you can freeze tissues or you can freeze fertilized eggs for a long period of time. And the idea behind this is this nutrient broth that they're in is very supportive. And then when we freeze them fast, really fast, so that there's no ice crystals and no damage. The way I like to think about this is you put some bacteria in the broth and you put them in the freezer and they're like, wow, it's getting really cold. And they just freeze, okay? And then you can pull them out a week later, a month later, a decade later, put them on a petri dish and they go cold, and then they like kind of look around like, oh, how are you doing? Yeah, they just like go back to what they're doing. Like they have no idea that they've been in this basically suspended animation. Okay, so they're like basically perfectly preserved. The only thing is we have to use special media for this that prevents frostbite and crystals, because the crystals that form, that's what can damage tissues or cells as it can pierce through them. So as long as they're in the special media, they're fine and you can keep them for a long time. Another way you can preserve bacterial cultures is to make them like freeze-dried ice cream. You do lyophilization. This is where we grow them, and then we put them in a vacuum chamber and quickly pull the water out of them. Now again, what's left behind is their cell remnants without water. It looks kind of like powder, but if I add water back, it rehydrates them, and they're able to recover. Like bacteria are pretty amazing as to how much abuse they can take. And this will work with probably, I'd say, about 70% of strains. There are certain ones that this won't work on. So as you can imagine, just like we had our mycobacteriums that we grow, in media, those princesses won't survive this. They're not going to be able to survive being freeze-dried. If a space station in orbit suddenly ruptured, let's say it blew up, the humans on board are died. Well, this sounds like a bad movie. There's cold and vacuum of space is present. Would all of the bacteria also be killed? No. So this goes back to your question, Aaron. They would still survive. They technically can survive in space indefinitely. They're basically going to be like we've just frozen them. They're just like, in space. The problem is getting them back. Because again, if they try to do re-entry, they're going to probably burn up. They're probably not going to survive that. But there is a whole group of scientists that believe that the life origins on the planet probably happened through meteorites. That when there was an early Earth and the atmosphere was different and maybe it wasn't as hot for entry, that a meteorite crash landed on the Earth and some microbes were on there and that started our first primordial soups. So that's one of the hypotheses that's been thrown out there. again, nobody's sure. There's lots of hypotheses. So many bacteria can survive without oxygen. They can survive in that cold, especially if they're an endospore former. Their endospores are fine. They're just like chilling out there. Same with viruses. Yeah, go ahead.
So is that like kind of the case when it comes to this deep freezing and localization? Is that it's not necessarily the bacteria themselves are being frozen, kept alive, but like endospore forming bacteria are more likely to survive this kind of stuff? And
Yes and no, depending on the process. So the freezing at that rapid cold temperature, that actually, it preserves it pretty well. It's
it
kind of
just stops metabolism.
stops everything, and if you have them in enough of that rich fluid, it actually doesn't allow cell damage. It's pretty amazing. So it'll actually just freeze them in place, no endospores at all. They'll be able to come back. But again, it depends on how princessly they are. Sometimes if they don't have certain enzymes or they're not happy with the fluid, then we need to have endospores. It's a good question. It really depends on the microbe itself. Okay, let's talk about growing in bacterial cultures. So binary fission is going to be the way that all of our prokaryotes are going to reproduce. We're going to look at a couple slides with binary fission, and we're going to see it's a pretty quick and easy process. This is why sometimes the bacteria can grow faster than our immune system can destroy them, and that's when we end up getting sick. We're also going to see that cells can undergo mitosis. That's what we do. Or budding. We're going to see that that's what yeast do. They can have chlamydiospores, those are special mold spores, or they can have fragmentation, where like things like mold fibers will break off and they'll reform. So let's visit each of these. First of all, this is binary fission. Binary fission is really the quickest way for a cell to reproduce, and archaea and bacteria will both do this. What's going to happen is they're going to get a signal from their DNA that it's time to replicate. And so what they're going to do is they're going to make a copy of their DNA, and when they make their copy, even if I'm a cox cell, I'll do the same thing, like a circle shape. I'll put my DNA on one side and my new copy on the other. Okay? And then, really quick, I'm going to grow a cell wall right in the middle, and I'm going to split my cell in two. Now, if my cell wall stays attached, this is how I'll build things like my staff, Coccus break clusters. I'll make another cell here and another cell here, and instead of separating the cell walls, they just stay attached together. And I make my little cell cluster. Or if I have diplobacilli, they would stay attached like this. Okay, so this is how I get the different cell arrangements. In this case, this is E. coli, who's a single bacterium and a single cell, so they separate completely. Okay? This process, replicating the DNA, building a cell wall and pinching themselves apart, takes maybe 20 minutes at the most. There's some bacteria out there that can even do this at 15 minutes, which means that every 15 minutes I get replication. So I go from 2 to 4 and 4 to 8 and 8 to 16, and you can see where it starts to get crazy. So if our immune system isn't on it, if it hasn't detected these guys, before we know it, we have a problem. We have a lot of bacteria cells, and now we got to try to get rid of them. Budding. Budding is where we have yeast cells that undergo mitosis just like us. Now what's funny is these yeast cells can do this process either sexually or asexually. So they can clone themselves and do this process, or they can mix genes and do this process. It's kind of funny. Either way, they're going to go through the process of mitosis, which we'll kind of cover on a few slides. And what's going to happen when they're done is they're going to end up with their daughter cell attached to them. So a budding yeast is where I go through the process of mitosis, and we're going to see that in the next few slides. But at the end of mitosis, our cells go through a separation. Our cells pinch off and separate. Yeast, they're like the helicopter moms. They want to keep their babies close. So they're going to hold on to them a little, and we're going to call that budding, and the daughter cells will stay attached to the moms maybe their entire life, or if they do separate later, they're going to both be adults. I love this idea. I tell my 13-year-old, let's be budding buddies. You know, I don't want you to leave. She's 13, she keeps leaving. I don't like it. Get over here. Yeah, so the cool thing is when you look at budding cells on a microscope, they kind of look like bowling pins. for like repair. If I get a cut, those cells need to make new baby cells so that they can fill that cut in. Okay, so I use it all out of damage control. Now, with mitosis, it works great, but I have certain life expectancies attached to each of my cells. So, for example, as we start to age, what happens to our skin? Starts to sag and wrinkle, right? The reason for that is when we're young and life is great, our cells are able to go through mitosis really quickly and our skin stays fresh and young. And then as we get older, their mitosis patterns slow down. And they're not creating offspring as quickly or at all. And so we start to have skin kind of bag and fold. That's what we're seeing. We're seeing aged mitosis. And we slow down. Okay, for cell division to occur, I have to do a lot of preparation. And I'm going to do most of my preparation in the stage that's referred to as interphase. Does this sound familiar? Remember interphase? I see lots of nods, yeah. So interphase is the stage where the interphase is the stage where eukaryotic cells spend most of their lives. Interphase means I'm just doing my job. I'm just doing my function. Y'all right now, you're being students. You're in interphase. You're showing up. You're taking notes. You're taking tests. You might be working. That's another part of your interphase. Okay, you're just living your life. All right, and then at some point in your life, you might decide that you want to have offspring. So then you're going to be going through a different stage. All right. So in interphase, that's where I'm going to get the signal that it's time to replicate. And the first thing I'm going to do is I'm going to duplicate my DNA. Now I'm going to do this in interphase. It's kind of like planning for a trip. Let's say I want to go on a trip. Do I just walk out the door and leave? No, I have to do a little pre-planning. That's what interphase does too. It's kind of like, okay, let's pack the clothes, let's iron things, and whatever we got to do. We're going to make some extra DNA. We're going to make some extra organelles. We're getting ready for the trip. Okay, so interphase is our prep stage. Then we're going to go into the process of actually mitosis, which actually happens pretty quick. That process is where we go from one stage to another, and then when we're done, we end up with a daughter cell. And then that's the end of mitosis, and then what we'll do is we'll go to cytokinesis. Cytokinesis means cell splitting. So that's where we're going to physically separate the daughter from the mother cell. So let's look at our process. So we start here in interphase. In interphase, we're going to make copies of our DNA. Then we're going to start going in and getting ready to go through mitosis. When we go through mitosis, I'm going to create a daughter cell, and then I'll physically separate them in cytokinesis. All right, so we're going to look at this process a little bit more. Now, to make a copy of my DNA, one of the big questions that researchers had to figure out was how was DNA formed, or what did it look like, and also how did it replicate? One of the big questions was, does my DNA molecule that looks like a double helix, does it make a whole new double helix, or does it keep some of the old strand and make a new strand? And through trial and error by tagging molecules, we were able to determine that we keep an old strand and build a new strand. That's brilliant. That's basically like giving yourself a template and you're just copying that template. That's pretty smart. That's a smart way to do stuff